So we're picking up chapter 5 again. The caption says, embarrassing moments at gene parties. And these guys are supposed to be chromosomes. Oh my goodness, Vinny's mutating. Turns out that a mutation eventually became a term applied to any change in DNA. So it could be a tiny change along the sequence of the code, which is the most common type of mutation. It could be pieces missing, pieces added, pieces flipped over, whole chromosome changes, whole sets of chromosome changes. So anything from the very tiny up to the very large, as long as the DNA you get isn't the DNA it was supposed to be, it's a mutation. Just to give you some idea also of how this gets way more complicated than we're dealing with here. Here's a situation where skin color is being determined by, there's 3 pairs here. So there's 3 different genes at work. And so whenever sperm and eggs are made, they're only getting 3 of whichever 6 they started with. And this is all the different mixtures that you can get in a multiple gene trait. And it's a relatively simple multiple gene trait. And this is why you see a lot of variation in skin color in the same family, whereas eye color is a little bit simpler. Eye color tends to be one major gene working. There's a couple of other ones at play that can shift things a little bit. But anything that's produced by a bunch of different proteins working together, you're going to get patterns that are, to some extent or another, predictable. You take a genetics class, it's very, very mathy. But it gets very, very tricky. You're just basically reducing to odds. What are the odds of getting a very dark color? What are the odds of getting a very light color? Well, we're continuing with the history, so I'm going to go back to where I was. So after it was discovered that viruses only needed to get their DNA into a cell in order to make that cell a virus factory, that looked like it was probably the code. But how does the code work? And then you get up into the 1940s and 1950s. Watson and Crick. Let me just make sure that's getting what it's supposed to get. Yeah. Watson and Crick, the nature of the DNA code, how the DNA code works. Now, these other 2 people worked in their lab, were incredibly important in developing the analysis that they needed in order to figure this out, but did not get the credit that Watson and Crick got. Rosalind Franklin, possibly because, well, probably because she was a woman, possibly because she was a Jewish woman. And I forget what the issue with Wilkins was, but Watson and Crick just went on to be, really famous based on their ability to portray DNA code in a way that made sense. So this is DNA structure, what's called the double helix. So when you break DNA down chemically, it's very simple. There's not a lot to it. And the question was, well, how can you get any kind of complex code out of something so simple? Now here we come to postmodernism. I'm not sure that Watson and Crick would have figured this out if they weren't in a world that had already developed recording technology. Recording technology where a magnet adjusts essentially rust on a medium. It started as wires and then it went to rusty tape. But the tape itself and the rust is very chemically very, very simple. But in order to get complex patterns of music and eventually images, it's the sequence. It's laid in a sequence and then it's decoded in a sequence in order to give you very complex information from a very simple system. And that's what they saw here. So the sequence of the nucleotides are a code for the sequence of amino acids in a protein. A protein is a long chain of amino acids. A gene is a long chain of nucleotides. And that sequence of nucleotides can be translated. It's transcribed first and then it's translated. That's why I paused there. You'll learn that in the second semester in a lot more detail. But it can be translated into the sequence that makes the protein. So this is basically how the DNA code works. And they figured out that you've got two strands, one of which has the code on it, one of which is the flip side. And when you need to make copies, you peel the strands apart. And the matchups are a predictable matchup. So as you're making a copy of this strand, the sequence on this side determines the sequence, because you're making another one that matches this one. Again, we're going to get into that in a lot more detail in the second semester. But it's a replication system, is that each strand can be pulled apart, and it's very easy to replicate the strand that used to be across from it. So you go from one piece of DNA that you're using for codes to two pieces of DNA that you now can pass on, one copy to an offspring cell and one copy you hang on to and keep using. Very, very, very important. So just to give you a preview, because this is pretty simple, turns out there's only 4 nucleotides in a DNA code. There's adenine, that's A. There's cytosine, that's C. There's guanine, that's G. And there's thymine, which is not the same as thiamine, which is a vitamin, which is T. And if you've got a T on this side, it'll match up with an A on the other side. So if you're making copies, wherever there was a T, when you're making a new copy, you're putting an A over there. That's what used to be over there. That's what's going in there as you're making copies. A G will match up with a C, and a C will match up with a G, and an A will match up with a T. When I learned this, the pairing was pretty easy. What I did was I put it in alphabetical order like I had it up there, and the insides pair up and the outsides pair up. It gets a little bit more confusing in the first step, which is transcription, where the DNA code is actually transcribed into an RNA code. And RNA has A, C, and G, but it has a U instead of a T, but the matchups still work. I am going to list this just so you know what they look like. Adenine, cytosine, guanine, and thymine, or thymine. So people started to understand that there's a code in DNA that matches the sequence of proteins. They'd already been pulling proteins apart and figuring out their amino acid sequences. Now they wanted to figure out how the sequence of DNA matched up the sequence of amino acids. And what you wind up with it's a 3-to-1 code. As you're going through the gene sequence, every 3 nucleotides is a code for 1 amino acid in the protein. And again, we'll hit that in a lot more detail in the second semester when we're dealing with molecules. But just as a simple layout of how this works, it turns out that the same amino acid can be coded for by a few different codons. That makes the code what they call robust. If you fiddle with it at a spot, you change one nucleotide to another, very often your protein is going to give you the same amino acid that it had. You change from one codon for, say, aspartic acid or aspartate to another code, gives you aspartic acid also, or gives you something that acts very similarly in the string. So again, it's a robust code, is that you can kind of chip at it and get the same thing that you would have gotten anyway. And this is all stuff that they discovered farther down the road. Another thing that was discovered about this time, actually a little bit later, when we talked about dominant recessive genes and alleles. Alleles are the variants of the genes, remember. Very often diseases are caused by an allele whose code produces a protein that doesn't work right. Very often doesn't work at all. But there are genetic diseases like that that are relatively common in some populations. For instance, sickle cell, which is a variant gene for hemoglobin, the oxygen carrier of the blood. I think I've gone through this before. I know I did in another class. At some point I get confused over what I've covered where. Normal hemoglobin is a nice round molecule. Oxygen sticks on it, nice round molecule. Oxygen comes off of it, nice round molecule. Very easy to package in nice round red blood cells. Sickle cell hemoglobin is only round when there's oxygen stuck to it. When the oxygen comes off, the protein starts to change shape. And there's actually 4 spots where oxygen attaches. If all 4 spots lose oxygen, the protein actually changes shape, and it goes from being nice and round to being long and pointy, slightly curved. And in a red blood cell, it can actually make the red blood cells curved, like a sickle head. That's where the name comes from. And those types of cells do not go through little bitty blood vessels very well. It's a bad, sickle cell anemia is a bad disease to have. But it is relatively common in African American populations. It's relatively common in some African populations. And this is where the first hint of what was going on was derived. I don't know who these people were. I read about this years ago and I didn't write the names down. But the story, as the story is told, is you've got 2 professors on the same campus, both doing their own research in their own areas. One's an African American studies researcher, the other one is an infectious disease researcher. But they're on committees and they deal with each other, and one day one of them's in the other one's office. I don't know which one's standing and which one's sitting, so I'm gonna make this up. So the standing guy is talking to the other guy about committees or whatever, and he kind of glances down at the guy's desk and goes, why do you have a map of malaria on your desk? And the guy looks down and goes, that's not what this is. I'm studying sickle cell in African American populations, and what I'm trying to do is trace this back to the parts of Africa that their ancestors were taken from. And that's what this is a map of. It's a map of the areas of Africa where these particular African Americans back 3-4 generations came from. It also matches a malaria map. Yeah. Wonder if that means anything. And that was the first step toward developing a theory of hybrid vigor, which I'm going to write down right now. I'll make my ones clearer. So sickle cell anemia is a recessive allele that exists in African populations, African American populations. It's a code for a not quite working version of hemoglobin. Now, if you carry one bad and one good, you have a sickle cell allele and a regular hemoglobin allele, you use both of them. Your red blood cells have a mixture of sickle cell hemoglobin and regular hemoglobin. But except in extreme circumstances of oxygen deprivation, you don't wind up with enough sickle cell protein changes to really affect your cells. If you go to high altitude, or you exercise really, really vigorously, or you get anemic for other reasons, and whatever drops your oxygen levels, it can be a problem. But typically, people with sickle cell trait don't really show any kind of bad symptoms. But that doesn't necessarily tell you why it would spread in generation after generation after generation to become somewhat common in a population. Here's what also happens. If you have sickle cell and regular hemoglobin mixed in your red blood cells, malaria at one of its stages, we'll talk about this later in the semester, invades red blood cells to reproduce. And when it's doing that, it depletes the oxygen levels in the red blood cell. When you deplete the oxygen levels in these red blood cells, they crinkle up. They don't necessarily form sickles, but they crinkle up. And here's another, you need to know this about red blood cells. They have a life expectancy. They wear out. And your system is set up to grab worn out ones, take them in, recycle the iron and a few of the other things, but just tear them apart to recycle them. If you are infected with malaria and you have one sickle cell allele and one regular allele, your cells crinkle up that have been invaded by the malaria. They are seen as expired, and they're taken in and pulled apart, and the malaria is killed. So you do not suffer from malaria anywhere near as bad as somebody who has either of these. If they've got two good hemoglobin alleles, then they're much more likely to die from malaria. If they have two sickle cell alleles, that's also bad. But the hybrid, one bad and one good, actually has a fitness better than either pair. Remember, fitness, your ability to deal with conditions, the conditions here being malarial parasites, is, the fitness is positive with the mix. So what it does is it holds and spreads the sickle cell allele, even though getting a pair of them is bad. Getting just one of them is very useful any place where there's malaria. No longer a great thing in the United States. We haven't had malaria around here in a long time, but it persists because one and one doesn't tend to be bad. And so, yeah, it's probably gradually being weeded out of the population every time you get two sickle cell together, although we do have treatments for it. Anyway, they found this for other things as well. Other genetic diseases, very often you can find a hybrid vigor example. Tay-Sachs disease seems to give a resistance to tuberculosis. I'm drawing a blank on some of the other ones. Even multiple gene traits. There's some indication that a combination of alleles that would give you schizophrenia in other combinations can make you artistic, very creative, maybe more of a risk taker, which is a useful thing to have in a human population. So in some mixes, it can have very positive fitness effects, and in other mixes, it can have very negative fitness effects. Still on the history. People started to find, if you knew how the code worked, and you could take a stretch of DNA and look at it, that an awful lot of time it was not a protein code. We will talk about that much later. That a tremendous amount of our DNA is not codes for proteins. It's what they originally called junk DNA, which is humans going, we don't know what it does, so it must not be doing anything important. It does do other pretty important things. But it does turn out that once you know what you're looking at, you can start to recognize stretches of the overall DNA, the genome. as gene sequences and sometimes other types of sequences. And so it was like, well, you know, it'd be really great if we could just get the whole thing. Let's get all of human DNA and have it all laid out in sequence, so that then we can start looking for genes. We can start to match protein sequences to DNA sequences and figure out where this protein's gene is and that protein's gene is. But we need the whole thing laid out first. And determining the sequence of DNA was a very slow, very labor-intensive job. But the National Institute of Health said, tell you what, the first group that can give us, I forget the threshold, like 90-some%. They didn't figure they were going to get 100%, but they wanted a lot. You know, a certain threshold of human genome would get this big cash prize. And they figured this is going to take 20-some years. And people started to come up with ways to automate what used to be done by human beings. And the prize was given out, I think, in a dozen years, but they actually had a lot of this new technology worked out within 8 or 9 years. And then they had the human genome, which is really the genome of just one person. And now they look at genomes of, you know, other people. And when you do like your DNA testing, if you go off to Ancestry or 23andMe, I'll talk about this on the next video, but they're not looking at your whole genome. They're looking at select parts, because the whole thing is just too overwhelming to compare. But once this technology was developed, people went, well, great. I've got this other living thing that I would like to know the genetics of. I'm going to use this technology to do E. coli genome, or dog genome, or chimpanzee genome, or, you know, monarch butterfly genome. Is that a tremendous number of genomes have been determined since the Human Genome Project, using the technology that was developed and then refined from the Human Genome Project. So this is a tremendous step in genetics history. And we're gonna, at this point, kind of leave this history list, and the rest of the chapter is gonna be more about particular details about, I mean, there is gonna be one more thing on the history list that's gonna lead to some other stuff. But I'm far enough in, I'm gonna stop this here.